final speaker of the day, Martin Turner. Martin is a Senior Research Fellow of Green Templeton College. He's Professor of Clinical Neurology and Neuroscience at the Nuffield Department of Clinical Neurosciences and a Consultant Neurologist at the John Radcliffe Hospital. And his talk is on measuring up to motor neurone disease.
So, I'm going to be talking to you about a neurodegenerative condition called motor neuron disease, which I'm sure most of you will have heard of or possibly even know someone with. I'm going to start off by showing you a video.
Hi, my name is Patrick. I'm a tree surgeon. I started doing tree surgery because I did a night class at college. I wanted to do Welsh, but they didn't have a Welsh course, so I saw tree surgery on this. I thought I'd do that, and I've just been obsessed with it ever since. The trees are just fantastic. I just got obsessed with them. They're just huge plants.
Hello.
So, motor neuron disease is, I think, one of the most really devastating conditions that I've experienced in the whole of medicine, really. We're used to the kind of image of some very aggressive forms of cancer, but this is a condition which takes someone in apparently normal physical health often to the point of complete collapse within 2 to 3 years of the onset. And as I'll sort of mention throughout the talk, really, this is not a condition that has some particular lifestyle or risk factors that we understand. The vast majority of my patients are notably physically extremely fit and athletic, and it can happen to anyone. I will touch on some of the genetic causes, but this is predominantly a sporadic condition, similar instance all over the world and in all sorts of socioeconomic circumstances, and can happen to anyone. So what Charcot described was this loss of the architecture of the nervous system, which you can see in the middle there. And the way we send messages to our muscles is from electrical activity in the brain down what we call the central or upper motor neuron tract in red there, and then out to the various components, arms, legs, speech and swallowing muscles, and respiration as well. And they're the lower motor neurons. And we divide that up as a sort of two-part circuit, and what's happening in motor neuron disease is that these are disintegrating jointly, but to a variable sort of dominance, upper or lower motor neuron. And actually many neurological diseases tend to either affect the central or the peripheral nerves, and motor neuron disease is unusual in affecting both. And it produces this pathological entity of muscle wasting, which is called amyotrophy, and stiffness and spasticity, which is called lateral sclerosis in the cord. And so the term amyotrophic lateral sclerosis, or ALS, is the preferred term that you'll hear internationally, but I'll use it interchangeably with motor neuron disease because it's all one thing. Now, the first thing people think of is, why did Stephen Hawking fortunately live for so long, and long enough to be able to contribute far more in terms of physics, and that's how he very much regarded himself, really, and the motor neuron disease was a sort of separate entity. And the answer is that we know that this condition has a variable prognosis. Once you get your pace set for you as an individual, it tends to stay the same, however. So if things are moving fast, they will stay like that. And Lou Gehrig, who gave his name to the American version of the condition, had a very typical course and died within 2 to 2.5 years of onset. Stephen Hawking lived for 50 years, and it was a long time before he required anything like ventilatory support. So that wasn't the reason for him living that long. And in fact, in our modelling here that we did as a large group, a large consortium, if you put Stephen Hawking's details into that model, it comes out with a 20% predicted chance that that individual developing symptoms as he did at the age of 20 would live to 70, which he did. So it's not out there, but it's definitely an extreme. And for the vast majority, 50% of our patients, this condition is going to take them from physical normality to death within 3 years. Now, most cases will not come to us with a family history. About 10% will tell us that a parent has motor neuron disease, or a related condition called frontotemporal dementia, which is a different sort of dementia from Alzheimer's, but it's a flag to us that we need to think about genetic causes, specifically the areas you can see in red there, which is this chromosome 9 gene, which can cause motor neuron disease or frontotemporal dementia. And if we go to our patients who are telling us they don't have a family history, the so-called sporadic ones, and we test all of them, we'll still find these genetic causes in about 10%. And of course, there's obvious reasons: people who were adopted or perhaps didn't know their family, diseases getting hidden in the family, or parents dying at a young age, and so the gene wasn't apparent. But for the vast majority of people, we have no single genetic cause. So the model that we think is operating here, and this goes back to Richard Dole's kind of work actually, where you plot the log instance against the log age, and you get a multi-hit model which fits best with the idea of sporadic ALS. And in this model, we think perhaps 6 things happen over a lifetime. And for those people who are carrying a particular monogenetic cause, it's still not absolutely certain they will get the condition, and possibly from our modelling, 2 other things need to happen, 2 or 3 other things. And that explains why it's more likely, but why we also don't have what's called 100% penetrance. Now, we have developed the signature of MND, but the problem is I can only diagnose that signature on post-mortem tissue. But within the brain and the spinal cord neurons, we see these protein aggregates of a molecule called TDP43, and think of it like the amyloid that you might have heard of in Alzheimer's. These are abnormally placed proteins which tend to aggregate. Now, is it their aggregation that's causing the problem? Possibly. Is it the loss of function as a result of that? Possibly. Is it some acquired abnormal function? Possibly. And that's the great mystery, really. But I think the fact that we have isolated that as the key change in nearly every single case of MND ALS, I think has been probably the greatest molecular breakthrough of the last 20 years. Now, the problem in diagnosis is not whether the person has MND. Sadly, by the time a neurologist, and certainly by the time I often see them in my specialist clinic, it's extremely obvious they have motor neuron disease. And there are very few cases where I'm scratching my head unsure. The problem is it's taken a year for them to get to a neurologist. And not all of that is sort of procrastination on the part of specialists. It's actually failure to attend to the GP. And then actually, I think, to be fair, many studies have demonstrated there is a failure of general practice to recognise that it's a primarily neurological problem, and to think of more common things such as nerve entrapments or other musculoskeletal diagnoses. What are the key barriers to therapy? Well, we don't know who gets this. I will mention at the end that we're starting to get some clues in that. We do have the delayed diagnosis, but that has multifactorial reasons, and the problem is patients have established disability at that stage. Clinical heterogeneity and this variability in rate progression is a big issue for us in trials. But the real problem is that we have to do placebo-controlled trials at present, and if we use survival as a marker, they're very long trials. And so we have this problem of measuring change. It's very easy to see what happened to that patient in the video I showed you, but the reality is that day to day, people don't feel different, and actually people with motor neuron disease don't feel ill in the same way as cancer patients. They actually feel extremely well throughout their condition. It's not a painful disorder, and it's not one that most people feel systemically unwell. So measuring change falls to simple functional measures, and they're notoriously inaccurate and insensitive. Now I seem to have temporarily lost control of the slides. There we are. So we've tried a lot of drugs. You don't need to read the list here, but all of these drugs have been tried in both in vitro, and some of them in capitals there have been tried in patients. And the ones in red there do have very small effects on survival and disease slowing, but really very, very modest effects indeed. And these drugs have taken typically 12 to 18 months trials to test them each time, and represent decades of work and decades of failure. The problem is not that we don't have candidates. I've got about 40 or 50 things that have shown some interest in both phase 1 and phase 2 studies. The problem is how to test them. Now we can use option 1, which is to do a survival trial and to take a group of patients, give them the drug, give them placebo and see who's alive at the end, and that will take 18 months to get a result. We can also look at their functional rating score, so-called ALSFRS, and that's on the right-hand panel here. And you can see that the typical people in the trial will have a whole range of trajectories. And so we take a mean of their decline, and we're looking for a difference in the rate of decline. And again, that's going to take us 12 months to get that result, and it's going to be fairly insensitive as a measure. Our patients are meanwhile trying lots of things themselves. There's a movement in the US called Right to Try, which has caused some difficulties and raised all sorts of debates and conversations. But the reality is patients will try all sorts of things, and I see that my role is to try and help them navigate that and try and help them get some sense, an objective sense of whether it might be helping. We do have some patient facing websites like ALS Untangled, where we help people try and make sense of the whole range of things that they get offered online. So I'm in the business of trying to find biomarkers, and broadly these are to get people into trials earlier, to stratify them into who's already we can see is in a fast or a slow track, so that we don't expect necessarily the same effect in a Stephen Hawking patient as we would expect to see in a Lou Gehrig patient, and we model that so that we're being fair in the assessment of the drug. And also particularly to try and find something that's more sensitive to change, even if it was something that we could just say, look, we've measured this, it's not changing over perhaps 3 months, and so let's try something else. Biomarkers also help us unravel key mechanisms. So what you've got there is a picture of a motor neuron with lots of things going on inside the cell and in the very long axons that supply the muscles. And you've got cells called astrocytes and microglia hanging around, supporting and pruning all the neuronal network. And there's a whole range of sort of headline mechanisms that have been discovered in cell biology: mitochondrial dysfunction, oxidative stress, changes in nuclear cytoplasmic transport, RNA processing, axonal transport, excitotoxicity, and all of those have their proponents and people who work on them. But the point is that they're a bit like the people crowding around that elephant there, and the concern is we really tend in science to grab one thing. And it seems very clear to me that neurodegeneration in particular is very unlikely to be one thing. This is a very complex set of processes that probably comes down to our basic lack of understanding of how a cell ages. Now, we've got various tools. We can look at the brain and the brain stem using MRI scanning and something called magnetoencephalography. We're able to look at the major motor tracks by stimulating them non-invasively with magnets called transcranial magnetic stimulation. We can look at the peripheral lower motor neurons by standard sort of electrophysiology, so-called electromyography. And increasingly, and I'll focus a little bit on, we can look at the body fluids, blood and spinal fluid. Blood obviously a lot easier to take. And there is some interest in urine, but so far it proved quite a difficult and complex biofluid to use. I set up a study some time ago now, and I suppose what was new about it and what we've built on was the idea of building something into clinic where we would take patients and study them longitudinally, and we'd study them every 3 to 6 months with a range of techniques and try to find ways to measure their disease. It's capitalized on a revolution in imaging, which I'm very pleased to have been a part of, and using MRI, magnetic resonance imaging, not just to get a picture. The sort of photographic benefit of MRI is obvious to most people, but actually it's to start to dig into the data of the scan and to look at the microstructure in techniques so-called diffusion tensor imaging, and also the function of the brain and how various parts of the brain are simultaneously talking to each other and how those networks disintegrate. And broadly speaking, very early on, we were able to demonstrate that we could see changes in the key parts of the brain in living patients. And what my colleague at the time, Gwen Dewar, who I think is on the call actually, it demonstrated by putting alongside the original 1960 images of the post-mortem slices of brain, we could see that we were seeing the same things in living patients: the damage but in a non-invasive way. And indeed, we can use this to start to track damage over time. When we do that, we can see there's a huge amount of damage in yellow. You can see in the top panel here, patients at baseline, showing us that although they may have only had symptoms for a few months, we can already see that neurodegeneration is the sort of process that is going for a long, long time before the symptoms emerge. And this is one of the great challenges. We have a lot of redundancy in the brain, and so by the time we have symptoms, it's often a huge amount of damage has occurred. But we can track some of that damage over time, and that's what you're seeing in the lower panel, where the so-called grey matter of the brain becomes more involved as we watch the disease progress. What about the fluid markers? Well, we can look in spinal fluid and blood, and we can start to look for markers of neuronal damage. And I think the most exciting of these is something called neurofilaments. They're not new and they're not specific to motor neuron disease, but you will get raised neurofilaments in the spinal fluid in the blood in any condition where you've got a high loss of neurons. And so it lends itself very well to a condition like motor neuron disease where we're getting a lot of change. We don't see it, the levels quite so raised in things like Alzheimer's and Parkinson's because they're playing out over a much, much longer period. But for motor neuron disease, this is an extremely promising marker. We can already tell that in the clinic, if it's raised, it's very, very unlikely to be a false positive. It does have some limitations when it's not raised. It can still be motor neuron disease, but that's a relatively small group. So we've got some real interest in using this as at least an adjunctive marker in the clinic. But its real benefit is as an intensity marker. So it tells me how active someone's disease is. And that's a huge advance for us because you can see in the panels below that the slower patients, intermediate and faster patients, the level reflects how fast their disease is moving in terms of their disability. The histogram at the top is just showing you that the very highest levels that we see in neurofilaments are in red there. We can see HIV, dementia, ALS and Creutzfeldt-Jakob disease are where we get the very high levels. And in other conditions, MS, Parkinson's and other conditions, it's much lower. What this allows us to do is to start to use neurofilament in trials. And this is just simply showing you that it's already being used as a marker of success in an MS study. So patients who responded to the drug with MS dropped the level of their neurofilament, suggesting that the disease process was slowing down. And some data that you can see on the far right there is some really exciting data showing a new genetic therapy for a rare form of motor neuron disease. There's some proof of principle that the patients not only started to feel better in terms of disability, but actually it was lowering this marker of neurofilament. There are other markers. We're very interested in the immune response. Is the immune response helpful in motor neuron disease? Probably in part. It may hold some of it in check. Is it primarily driving the disease? No. This is not a primary immune disease like multiple sclerosis. For example, bone marrow transplant does not help motor neuron disease patients. But these markers are very helpful to start to put into panels so we can look at the contribution of both the immune system and other factors in terms of disease speed. So just to wind up, how are we translating this? Well, we used the money from the Ice Bucket Challenge to fund a project really to start to roll out sampling patients in clinic across the country, and starting with 3 centres to do this routinely. And it involves a lot of practical logistics, really. The team has to prepare a huge range of samples, as you can see in the top left there, and tubes associated with that. And then down in the clinic, a much younger version of me there is persuading patients. It doesn't take much persuasion, but persuading people to add on a bit of extra time to have these research samples taken. They have to be processed, stored in liquid nitrogen. And to develop that pipeline is really challenging. And we all know in our busy jobs in the clinic, there's 101 reasons why we can say it's just too busy to do that. But it's the only way we can really advance being able to monitor patients without having to drag them up for independent separate visits, which particularly at this time in this climate is just not going to be feasible. Whereas we can see patients face to face with COVID precautions and still take these samples. So the last thing I wanted to just tantalise you with is just this idea of when the disease begins, really, because we tend to place our drug in at this part of the disease. This is a tablet of riluzole, and we're putting that in really when the disease is finally manifest. And as we know from all systems in the body, the redundancy is huge. And so there's this huge area of loss of tolerance and decompensation that's been occurring with neuronal loss, possibly over decades before. And we really like to give our drugs much sooner. And the way we can look at this group is to take people who have a genetic cause. They're a small group, just to remind you, but we can go to the families where we have the very high penetrance mutations, such as the chromosome 9 mutation. And we can say to relatives, look, would you like to take part in research? You're a healthy person, and you may or may not be a carrier of the gene. And we have a process whereby we can test them. Very importantly, I can stay blind to that result, and even more importantly, they can stay blind to that result. And then we can ask a third party to separate those into carriers and non-carriers for us anonymously. And then we can analyse the data and start to look for the very earliest changes in people who have no symptoms. Now, when that's done with those very sensitive imaging techniques I mentioned, this was a French group who have done this in particularly young people who are carriers of this mutation, highly likely to develop motor neuron disease in 20 years' time or less. And they already have changes in their structure and functional brain scans, which would certainly be the sorts of measures that we could start to think about using if we're going to invoke preventative therapies. In our own work, we've used functional measures, and what you're seeing on the left-hand side here is using the MRI scanner to come up with a metric really of the way the brain communicates with different parts of itself in something called functional MRI. And broadly speaking, you can see that the pattern of change in the affected patients, people with symptoms, are in the red dots there, and healthy people of a similar age are in the green dots. And scattered sort of in between the two are these pre-symptomatic carriers who have no symptoms, but you can see that the pattern of functional changes in their brains is sort of heading towards a halfway house, really. It's not quite normal, but it's not quite as bad as the affected people. And similarly, on the right-hand side, you can see some changes across the brain. We're looking at the top of the brain there, using the magnetoencephalography to measure changes during movement. And we can see the typical pattern of activity in the motor cortex in the affected patients, but the hint of some early changes in people who are carriers of genes, very likely to get the condition, but currently have no symptoms. And lastly, we've got some very interesting data coming through now looking at GP records in the Swedish Morris cohort. The dotted line represents the patient, the normal population who do not develop MND, and going back 20 years, the solid line is people who retrospectively turned out to develop MND. So in a population denominator of about 6 million, maybe 600 of those patients developed MND later in life, and the GP records were available to look at various blood tests throughout their life. And what it's showing is that there are quite significant divergence of certain lipid profiles and other markers of metabolic function 10 to 20 years before those people go on to develop MND. And it really suggests that there may be some quite profound changes in metabolic function and as well as in neuronal function. And we're also very interested in whether that might relate to things like the hypothalamic axis. And we can see there on the right-hand side that we even do see some changes in the size of the hypothalamus in the brain in people, again, who are genetic carriers. So I'm going to leave it there. What I've hoped to have done is given you a flavour of that this is a difficult condition. I have to say I'm more optimistic now about treatments than when I started, so that's good. But neurodegeneration is a very tough nut to crack. We're not dealing with a single system that can be switched off with a single drug. This is a complex problem of probably initially development, but then cellular aging over time. And I think the way we solve these problems is to take our patients in our clinical research settings in the clinics and involve them in research. I'm very grateful for the team which is shown below for helping me do that. I'll take any questions. Thanks.
Thank you, Martin, for that very interesting talk. You mentioned just a few numbers in relating to prevalence in the Swedish Amaris cohort, but what is the prevalence of this disease? I tend to think of it as something that's very rare, but clearly it's not as rare as I think it is.
No, so if we talk about the incidence, it's actually not that dissimilar in instance to multiple sclerosis. It's a little bit lower, but and you know, I would say most people kind of know someone with motor neuron disease. So it's but the prevalence is lower, of course, because the survival is less. But at any one time, there's about 5,000 people in the UK with motor neuron disease. So it's certainly not an extremely common condition compared to stroke. But it's more common than people realise.
Yes, right. I've got a question from my predecessor, Denise Livesley. She says, what suggestions would you have for improvements in the speed of diagnosis? For example, how might GPs be helped to identify possible cases earlier?
Yeah, so I think at grassroots level, and I think Lawrence is on the call, Lawrence shares my passion for neurological teaching at undergraduate level, and I think it's a very strong feature in Oxford. So I do think we know that when you interview most GPs in practice and ask them about neurology, they say it's the thing they feel the weakest about and the thing they found most difficult at medical school. So that's the start. Then I think the Motor Neuron Disease Association has also given a number of useful resources to GPs with the sort of basic mantra of painless progressive paralysis in an otherwise sort of fit person, and just to have that threshold, just to think, well, okay, could this actually be a neurological problem? So I think that helps a little bit, but I think we shouldn't fool ourselves that that we're going to be able to be seeing someone with motor neuron disease within 2 weeks of their first symptom. I think we can get it down from a year to probably nearer 6 or 9 months. But there is always going to be a delay because someone who's previously been completely fit will not assume that they've got a serious cause for their foot drop or their weak hand as the first thought.
And I've got a question here from Chris Winchester. It says, based on what you shared, is it fair to say that what is good for the heart is good for the brain in people at risk of motor neuron disease?
Yeah, I mean, the risk factors for some of the other neurodegenerative disorders, I mean, Alzheimer's obviously has a very strong thread now on vascular risk factors. I would say that, you know, I do notice, and Lindy and I have talked about this a lot, I do notice that my motor neuron disease patients are really strikingly athletic. There is some suggestion they may, as a result, possibly have a lower instance of cardiovascular risk. And I have to say, I don't think I can even recall a single MND patient who has subsequently, even in a long disease course, has subsequently developed heart or stroke problems. So I think probably there is a little bit of a divergence from typical vascular risk factors for motor neuron disease. And I'm more interested in how people who get motor neuron disease, how they process their food. You know, they often tell me they eat what they want and never put any weight on. And that we know that they're much slimmer. So I think there's an interesting story there about, but possibly divergent from the typical risk factors.
Okay, and there's a question from Paul Brankin. It says, my brother-in-law died of front temporal dementia 5 years ago. How great is the risk to his children of developing motor neuron disease?
Yeah, so the frontotemporal dementia is definitely, it's something that we've become much more aware of. And if I'm taking a family history in clinic, I wouldn't just say to someone, did your parent have motor neuron disease? I would say, did they have dementia? And if they say, oh, yes, they had Alzheimer's, I would be very, very careful in quizzing to check it wasn't frontotemporal dementia. A lot of frontotemporal dementia, but not by no means all of it. A lot of it has a more genetic basis. But there's only one gene that particularly ties it in with motor neuron disease. But it's the sort of thing that I think now there will be a longer conversation. The absolute risk to a family member is low, but I think it's the sort of thing that would be more routine to talk through with either a GP or a genetic counsellor, because frontotemporal dementia is a little bit rare.